Okay, so welcome to This Week in Cell Biology. Today we're looking at topic 16 on transcription. This is a very important one. So make sure you take a look at this very carefully, go over the notes, make notes, ask questions, do everything you need to do to make sure that you know. So let's talk a little bit about RNA. We've talked a lot about DNA. We're going to talk about RNA. You may know that RNA is RNA because it's ribose, and DNA is D because of deoxyribose. This has to do with the pentose sugar. Here are the two pentose sugars, ribose and deoxyribose. You can see the differences here, where the deoxyribose, deoxy actually means without oxygen, whereas the ribose has that oxygen at the 2' position. So it's ribose. What else is different about RNA? It's usually single-stranded. There are always exceptions in biology. RNA also has the nitrogenous base uracil instead of thymine. So if you take a look, the structures are very similar. Only difference is this methyl group here, which is just enough of a difference that RNA enzymes will recognize uracil over thymine. So here's the 4 bases, the nitrogenous bases that you're going to see in RNA: adenine, uracil, cytosine, guanine. Don't forget the other parts and nucleotides. We've got phosphates and riboses, and that's what makes up the nucleotides in RNA. So just a reminder, your final exam, you must know all these types of RNA. There will be a question on the final exam. I've mentioned this already in class. I'm mentioning it again. The question is going to have all 6 of these types of RNA. On the one side of the question will be the name, the other side will be the definition, function, and you'll have 6 blanks. So in some cases you'll have to put down the name, in some cases you'll have to put down the definition or what it does. So make sure you learn them. Today in this lecture we will be talking about messenger RNA, and we'll also be talking about small nuclear RNA. So take note. We've talked about the telomerase RNA 2 topics ago, and we have a couple more types to discuss. So RNA production is called transcription, and this is done by RNA polymerase. The amazing thing about RNA polymerase is it is a pretty amazing enzyme if you compare it to DNA polymerase. Remember, DNA polymerase requires all those accessory enzymes and proteins. DNA polymerase requires helicase, it requires single-stranded binding proteins, topoisomerase, it requires a primase, it requires the ligase. RNA polymerase does not need any of those things. It can separate the strands of DNA on its own, doesn't require a primer, is just ready to go on its own. So you can see here's a picture of the enzyme, RNA polymerase, and you can see the double-stranded DNA right here, and it's zooming along and producing this RNA. It's going to produce RNA from the 5' to the 3' direction. This is the direction all nucleotides are made in. So remember that 5' to 3' stuff. It is important. There it goes. Just a bit of an aside, there are different types of RNA polymerase. If you are a prokaryote, you only have one type. If you are a eukaryote, you have several types. Different RNA polymerases are responsible for different genes, whether it's messenger RNAs versus the transfer RNAs and so on. I do not expect you to know this for the final exam. Okay, so let's talk about this DNA. There's actually 2 strands of DNA. So we have to actually start to distinguish the difference between these 2 strands of DNA. First strand to talk about is the sense strand. So what is the sense strand? The sense strand is that strand that looks like the messenger RNA in sequence. So it has basically the same sequence. So the sense strand, of course, will have a DNA sequence, so it'll have T's. The RNA will have U's, but other than that, the sequence will be the same. So if you have, let's say, an ATG on the sense strand, the RNA will be a UG. But other than that, it's pretty much the same. So the sense strand is also where we're going to find the start codon, and the start codon is obviously very important. The other strand is called the antisense strand. I think that's what our textbook is using. I actually prefer the term template strand. So template strand, what does that mean? It means that this is actually what the enzyme, the RNA polymerase, is looking at. So it grabs it, it takes a look, and wherever it sees a C, it's going to put a G on the RNA. Wherever it sees a G, it's going to put a C on the RNA. Wherever it sees an A, it's going to put a U on the RNA. Wherever it sees a T, it's going to put an A on the RNA. So there's our little RNA that's being produced. The DNA on the sense strand is 5' to 3', and the messenger RNA is, of course, 5' to 3'. Like I said, the sense strand looks like. So something else to talk about with a gene. Transcription is basically done one gene at a time. You can see there I'm marking where the gene is found. And the language we say is what is before the gene is upstream, what is after the gene is downstream. So what is upstream of the gene? So upstream of the gene is something called the promoter. I have a definition of the promoter on the next slide, but basically a promoter is a DNA sequence, and this DNA sequence is where the RNA polymerase binds. This is where the enzyme binds. So it actually starts before the gene. So it's a little sequence that says, hey enzyme, come bind here, and you can start making my RNA. Of course, downstream we have something similar. It's called the terminator, and the terminator where the enzyme actually detaches. So it's a good idea to do this before the gene and finish after the gene. You get a little bit of extra RNA on there as you're producing your RNA. So the promoter and terminator. And there's my messenger RNA, which is going to contain a little bit before and a little bit after the actual gene. So there's the definitions. You can take a look there. You can take a look in the notes. Also, the textbook has a lot of information on these things. I will be using these words. It's very important with these topics that you do familiarize yourself with the vocabulary because it's important. Okay, so let's talk about RNA production or transcription and what is going on. So here's the images from the textbook. You can see the very first step is that the RNA polymerase binds. Where does it bind? It binds to the promoter. That is the portion before the gene. So like I said, that's a little sequence that says, hey, come bind here, start over here, RNA polymerase, and so it does. So RNA polymerase unwinds the DNA. It does this without the help of anything like a helicase. It does it all on its own. And there it goes, and then it's going to start to take a look at the template strand, and it's going to add a complementary RNA sequence, and it's going to make it in the 5' to 3' direction. Along a little bit, you can see there it goes. The RNA is being produced. So right here we have the RNA being produced in this direction. So it started over here. Start codon is probably somewhere around here, and it's producing, adding the nucleotides onto the 3' end, so growing in the 5' to 3' direction. It does this several base pairs at a time. It actually goes really quickly. This is a very, very efficient process. I mentioned complementary base pairs. The DNA reforms its hydrogen bonds after it gets separated, and after the RNA polymerase is done, the DNA is left unchanged. So there's kind of a zoom in, a little bit more detail. You can see the complementary base pairs. So down here on the bottom, you can see we've got a sequence here of nucleotides. So this is the template strand. And then the RNA polymerase is adding these nucleotides complementary. So this T is matched with an A, this A is matched with a U, this G with a C, this G with a C, and so on. So lastly, finally, it makes it to the end. That's where the terminator is. That's the sequence after the stop codon that says, hey, time to let go. So the RNA polymerase detaches, and that's the end.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix.
With this. Not sure what's going on with my
RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule.
So check out the video.
Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So what about eukaryote transcription? Let's talk about that for a minute. There's not a lot of differences. Most of it is exactly the same. One thing I want to point out is that promoters in eukaryotes often have this little thing here called a TATA box. So you can see TATA, T-A-T-A. And actually the complementary version is T-A-T-A as well, if we read everything from 5' to 3'. So that's one way in eukaryotes we often know that we have a promoter. There seems to be a bit more variety in prokaryotes, but eukaryotes often have this TATA box. So when you're looking for a promoter, look for this TATA box, and you might see it. So second thing that happens, so eukaryotes are a lot more complex, and we require something called transcription factors. So what are these things? Well, if you think about your body, how complex it is, you have cells that are blood cells, you have skin cells, you have bone cells, you have brain cells, you've got all sorts of cells. So first of all, we need to be able to turn on some genes in some cells, but not all cells. Secondly, we're multicellular organisms, and we start off as, we start off as babies, juveniles, fetuses, we grow, we mature, and so some genes need to be turned on or off during different times of development. So these transcription factors are tissue-specific or developmental stage-specific, and they're essential for the RNA to polymerase to bind before it starts producing RNA. So these transcription factors, like I said, they're just a little added piece of regulation because eukaryotes tend to be a lot more complex. So there's the transcription factors. You can see they're binding, and then there comes the RNA polymerase. So it's this big blob here. It binds, and it forms something with the transcription factors called the Transcription Initiation complex. So there it is. Other than that, it's just ready to go. It's going to start making the RNA. It's going to make it in the 5' to 3' direction, and it is ready to go. So eukaryotes are more complex than this. We're not going to really cover this, but you can read about it in the textbook. It talks about enhancers and silencers and repressors and all these kind of things, and you can see that they're all involved in the transcription regulation process, but we are not going to cover that this semester. Okay, so here a little bit more information about eukaryotes and their transcriptional process. You can see here's the gene, kind of similar to the diagram we showed you before. You've got the promoter before the gene, you've got the gene being transcribed here. After the gene is the sequence here, AAU, AAA, so you find that in eukaryotes a lot. Sometimes it's called a polyadenylation sequence because it's a bunch of A's, so poly-A sequence. And at this stage, we still call this the pre-messenger RNA. It means it's not actually messenger RNA yet because there's still some modifications, some adjustments to be made before it actually leaves the nucleus. So before I get to what happens with those adjustments and modifications, I want to point you to this video. This is a very cool animated video. I am going to add it to the playlist for this course, so go and enjoy that right now. Okay, so actually before we talk about those modifications, let's do one of these transcription translation problems. This is very similar to what we did before in the previous unit with some added complexities. So you can see I'm asking a few more questions here. So you can see the very first thing I'm asking here is identify the sense strand and antisense strand. So let's take a look. So there we go, the sense strand and the template strand. Remember, template, antisense. You can use either terminology. So how do I know that this one is the sense strand? Well, I'm looking for the start codons. Remember the start codon is AUG or ATG. So take a look along here for an ATG, and there it is, ATG. So there's the start codon. That's how I know that this is the sense strand. So good job. I know at least one or two of you are probably thinking, well, what about this ATG? Well, that's not actually ATG. Remember, we always read DNA in the 5' to 3' direction, so this is going to be read in that direction. This is GTA. So that would make this the template strand. Little hint, in my class, the sense strand will always be the top strand. I'm not going to make it confusing. I think it's going to be pretty straightforward for everybody. But always, always make sure you can find that start codon. That is always the place we're going to get started. Okay, next thing, give the RNA sequence. Okay, so let's do that. So the enzyme RNA polymerase doesn't start at the start codon. It actually starts before the start codon. Notice I actually put a TATA box in there as part of the promoter. So it's going to start somewhere over here. It actually starts a little bit before the TATA box. And so we're going to start over here. So remember what the enzyme does. The enzyme is actually going to take a look at the template strand. Where it sees an A, it's going to put a U. Where it sees a T, it's going to put an A. Where it sees a G, it's going to put a C. Or the other way is just to copy the sense strand. So TAC becomes UAC, always changing our T's to U's. Okay, our TAT becomes UAU. Our ACG becomes ACG, and so on all the way down into the end. So there's the messenger RNA. Question C. Identify where the promoter and terminator sequences are. Okay, so let's see here. The promoter, we've talked about this already. It's before the start codon. So circle or label before the start codon somewhere. We have to find the stop codon. We haven't quite done that yet. I'll get to that in a minute, but the terminator is, of course, going to be after the stop codon. And so I have that label there as well. Next question, give the amino acid sequence. Okay, so we start with our start codon. So there it is, AUG. That's our start codon. Now I'm just going to take a look and find all the triplets. So I'm just going one codon at a time all the way to the end. And there is no triplet here. I only have 2 base pairs, so I'm going to stop there. Okay, so the next thing I'm going to do is just start looking at the genetic code and translating this. So AUG, if you don't remember, is methionine. Always methionine. AUG. Start off with methionine for every single protein. UAU is a tyrosine. CCU is proline. UAA is the stop codon. So I'm actually done my amino acid sequence there. Remember, there's no amino acid named stop, so I don't need to write anything. I'm just writing it to remind you that this is the stop codon, but you do not need to write that. Okay, all good. So this is almost all you'll be asked for the question for the final exam. I will ask a couple other things about mutations. We haven't covered mutations yet. That will be in the next unit. So we will do another sample problem looking at mutations. Okay, so let's talk about that modification. We call this post-transcriptional modification because it's after transcription. So there's a few things being done here. So you can see here is the coding sequence of the gene. We have some things going on before the gene and some things going on after the gene. So before the gene, our cells add something called a 5' cap. So 5' means it's at the 5' end and it's a cap. Not sure who named it, but basically you can see it's a guanine and a bunch of phosphates, so 3 phosphates. So what does it do? First of all, RNA is a very sensitive molecule. It degrades very easily. It actually protects from degradation. Secondly, it signals nuclear export. If you remember, we were talking about nuclear pores way back in topic 6. We're talking about how some things go in the nucleus and some things go out. Things don't just go in and out just like that. They actually have to be labeled in the cell and be targeted to go in or out. So that's what the 5' cap does. Lastly, you can see it also helps, it says there it helps with ribosome binding. At the other end, we have a poly-A tail. So this is just a whole bunch of A's. This can be from a few dozen to a few hundred. Really, the longer the tail, the longer the messenger RNA. Some messenger RNAs actually last not even minutes. Some last a little bit longer. So the more A's you have on at the end, the slower the RNA degrades. Like I said, RNA is a very sensitive molecule, degrades very, very quickly. It also helps with ribosome binding, but this is mostly a protective measure at the 3' end. So there's the 5' cap. You can see what the structure looks like. You've got 3 phosphates. You've got a guanine here, and there's a ribose, and there's a methyl group here as well, if you're interested in the chemistry. Remember what it does. It protects it from degradation, signals it for nuclear export, and helps with binding to the ribosome. So there is our RNA, and you can see the next thing that happens is something called splicing. So take a look. There's the pre-messenger RNA up at top, and then we have our messenger RNA. So we've got our 5' cap, we've got our poly A tail, but notice the coding sequence here goes from being something very long to something being much shorter. So within the coding sequence, we have these two things. One of them is called the exons. Exons are things that are expressed, meaning those are the sequences that are actually used by the protein. Introns are the other region, and they're interruptions, meaning they are cut out and not in the final messenger RNA. So how is this done? This is done by something called the spliceosome. So let's talk about the spliceosome for a couple of minutes here. The spliceosome is basically this piece of molecular machinery. I don't want to call it a protein because it's not just made out of proteins. And this machinery is involved in splicing, so it cuts out those introns. So notice this note here. It says that it contains fragments of something called small nuclear RNA. So small nuclear RNA, snRNA. This is our next type of RNA, another type of RNA for us to talk about. It's called small nuclear because, well, it's small, nuclear, and it's found in the nucleus. So that's an amazing name, I know, but it's named that because, well, people discovered it and then eventually figured out what it was for. So this spliceosome is made out of proteins plus RNA. So it has two things. So it's not called a protein. It's not called a nucleotide. It's called it has its own name, spliceosome. So what is the RNA doing in there? The RNA is doing two things. It's recognizing introns, and it's also involved in the catalytic activity of this whole thing. So there's your definition. Small nuclear RNA is involved in the structural and catalytic components in spliceosomes. So in the diagram here, you can see we've got a gene with an exon, intron, and an exon. The intron is being cut out here by the spliceosome, and there's our final messenger RNA down there at the Okay, so what's going on here? Why do we have all this DNA or RNA? Like, why are genes so long? And why are we just cutting things out at the end? I'll get to that in a moment. But one thing that we've noticed with these exons is that each of them seem to form a domain in a protein. So if you take a look here, exon 1 forms this domain here in this protein. So what's a domain? It's kind of a part of a protein that is kind of independently folded, and it's sort of a module of that protein. So interestingly enough, a lot of genes have something called alternative splicing. So if you take a look, this is a gene called troponin. It's a protein. And we've got the DNA, and you can see it's got 1, 2, 3, 4, 5 exons. And so you've got that RNA being produced here. So here's the RNA. And so in this case here, you have two different versions of the protein being made. So version 1 has 1, 2, 3, and 5 exons being put together. Version 4, version B has 1, 2, 4, and 5. So this is called alternative splicing. So this is actually really quite incredible because now we have a method where we can have one gene, and that one gene can actually code for several versions of a protein. This is really quite remarkable. So we have 20,000 genes in the human genome, but the possibilities are much, much larger based on what we can do here with this alternative splicing. Some of these intron things we don't really understand fully. It's quite honestly something that might have to do with our evolutionary past. There may be other functions that we don't fully understand. Let's talk about this alternative splicing for another minute. So what I want to do is I want to talk about this alternative splicing and how it's used to produce something called antibodies. So antibodies are these molecules here. They're kind of Y-shaped. They're used by your immune system to recognize foreign invaders. This is kind of what an antibody looks like. You can see it's basically 4 proteins, 2 larger ones and 2 shorter ones that are attached together. And you can see over here we have this region here. It's called the variable region, and this is what So here's an example. You can see you've got an antibody here. Here's the antibody. It's recognizing a virus, and that virus is being labeled and targeted for destruction. You can see down here it is being consumed by an immune cell, taken in by phagocytosis, and then ultimately destroyed. So our body has a possibility to make many of these antibodies. You probably know that once you're exposed to something, you can get immunity to it. So sometimes we're exposed to things by vaccination, or sometimes we're exposed to things naturally, and we're going to get immunity to it. But we don't produce these antibodies until we're exposed. Of course, our body doesn't know what it's going to get exposed to. This is why it has the possibility to produce many, many, many more than 20,000 genes, many more than 20,000 possibilities. And we're going to do this with alternative splicing. So if you take a look here, this is showing what's going on with the DNA for antibodies. We've got many, many exons, and there's all this mixing and matching. One gene here you can see has exon 3 and 4 and 5 and 6. The other one is using only 3 and 4, but not 5 and 6, and many, many possibilities for antibodies. So this is one thing that we understand well for that alternative splicing. Okay, so some other footnotes on this topic of transcription is that there are differences between prokaryotes and eukaryotes. If you take a look, I made a table here looking at prokaryotes and eukaryotes and also comparing the bacteria and the archaea. You can see the first thing with RNA polymerases is bacteria have 1, archaea have 1, and eukaryotes have at least 3. Kind of depends on whether you're a plant or an animal or fungi how many you have. What about transcription factors? Well, bacteria do not have transcription factors, eukaryotes do, and some archaea do. So sometimes archaea do behave a little bit more like eukaryotes than the bacteria. What about post-transcriptional modifications? None or very minimal in bacteria. Eukaryotes, we talked about the 5' cap. We talked about the poly A tail and the splicing. Archaea sometimes have splicing, so kind of interesting. Okay, so that finishes topic 16. Like I said, this is a very important topic, so I'll let you read over these on your own time. You can download the PowerPoint as usual. Don't forget to go over the details. Be prepared to explain yourself in words on how transcription works. Also be prepared to do one of those transcription translation problems. Here's the terminology. Don't worry about ribozymes. We're not going to cover that in this course, but everything else there we did talk about. These are 2 separate things. Transcription initiation complex and splicing. The PowerPoint just kind of mushed them together a little bit. Here's some study questions. Again, I'll let you take a look over those on your own time. Hope everyone is doing well. Have a wonderful day.